The National Investor's motto is, you can get information anywhere, but here you get knowledge. And for well over 40 years now, Chris Temple has imparted that knowledge, an unparalleled, understandable, and useful take on the big picture of the markets and of our world. He has been out in front of what you see on the news every day on many themes. More, Chris has taken this macro knowledge and his unique analytical abilities to uncover unique investment opportunities. Often, he has guided his members to big winners in resources, biotech stocks, and more. His talents and connections often getting him into ground floor opportunities before they are discovered. Without further ado, here is Chris with today's topic.
Welcome back. In just a few minutes, I'm going to be joined by Michael Breen, who is the Executive Chairman and CEO of GT Biopharma. GT Biopharma, as you see here, is listed on the NASD under the symbol GTBP. Of course, he and or myself will very likely, I can almost guarantee it, be making some forward-looking statements, talking about what we hope is on the horizon to extrapolate from some recent good news from the company. So be mindful of that. As with a lot of biotech stocks, pretty much all of them, it's always speculative. I oftentimes liken small biotech stocks to small resource explorers. You need a lot of things to come together. You need the right people, you need the right project, you need money. And that hasn't always been in the same place at the same time for GT Biopharma. It's a company I became familiar with probably about 5 years or so ago. They had a nice run for a while after they got listed on NASDAQ, and then quite literally things started to fall apart. They had management issues. There's all new management these days. They had financial issues. And it was really true for a while that not only was the stock price broken, which did nothing but go down for the most part for the last 3 or 4 years, but that the company itself was fairly well broken. But as all of this was falling apart, Michael came in first as the interim CEO, then as the full-time CEO and executive chairman. He's bolstered the board in the recent past. And as you're going to learn when I bring him on, he is married now. Sufficient funding, by all appearances going forward, to match the world-class science that GT has, which none of us who have followed the company have ever doubted the science. In just a minute, I'm going to play a quick video that gives an overview of their science. Mike will get into more detail as we have our discussion. It was developed by Dr. Jeffrey Miller, who I had the pleasure of meeting and having breakfast with last summer when I was up in the Twin Cities. He is one of the best minds on planet Earth when it comes to cancer. And they're looking to do things better. Mike will point out that it's been decades that we have been pretty much in the US and worldwide, I think, for the most part, under the same kind of cut and burn care for cancer. And it's time for something new. It's time for something better that is less invasive, doesn't harm the body, and can help people suffering from all manner of cancers. Here in this video, before I bring on Mike, you're going to get a quick overview. It's remarkable science at the same time as being a fairly simple and straightforward idea to have a compound, which they do, and it's a whole platform of compounds that they're looking to get through trials and eventually bring to market that take your body's own NK cells, natural killer is what that stands for. It's a white blood cell. They supercharge it. They turn it into a serial killer. I love that term. And they do it without harm to the body. And this has had some early success in trials. They've got more work to do, but things are looking very good. And again, and I'll let Mike explain the details, now that it looks as though they're going to have sufficient funding to carry this forward for the foreseeable future.
GT Biopharma is a clinical stage oncology research company dedicated to harnessing the power of immunotherapies. The company is listed on the NASDAQ as GTBP. In the quest for innovative solutions to humanity's most challenging health issues, the emergence of novel protein technology holds immense promise. Our tri-specific natural killer cell engagers, or TRiKE for short, is a first-in-class protein therapeutic platform that enhances natural killer cells' ability to eliminate cancer. It improves ADCC activation, proliferation, and boosts NK cell persistence, and helps to reduce toxicities, thus creating a safer pathway to treating hematologic cancers and solid tumors, including prostate, breast, ovarian, and lung. By focusing on natural killer cells through our Trike platform, we aim to enhance the body's natural capabilities, creating immunotherapies that can specifically target and destroy malignant cells. Our senior medical doctor, Dr. Jeffrey Miller, is the inventor of the Trike technology. He is a professor of medicine at the University of Minnesota and director of the Masonic Cancer Center. With more than 20 years of experience in NK cells and immunology and over 170 peer-reviewed publications, he is a recognized key opinion leader in pioneering cancer research. GT Biopharma's goal extends beyond cancer treatment. By applying our novel protein Trike technology, the company aspires to develop therapies that not only treat cancer, but also address autoimmune and other disorders. Research has indicated that certain proteins can help regulate immune responses, offering potential breakthroughs in treating diseases like multiple sclerosis and rheumatoid arthritis. Ultimately, our mission is to improve patients' lives by treating cancer and other diseases in a safer, more humane, and natural way when compared with traditional options like surgery, chemotherapy, or radiation. With continued dedication to research and innovation, our vision remains focused on transforming the landscape of core treatment for cancer and beyond. GT Biopharma, immunotherapy for the treatment of cancer and autoimmune diseases.
And here is my friend, CEO and Executive Chairman, Michael Breen of GT Biopharma. Mike, good to see you.
Great to see you, Chris. Always great to see you. Always happy to be on and speaking to your audience of investors. So thank you for organizing this and having me on.
Oh, my pleasure. You know, unfortunately, you and I just narrowly missed seeing one another in person in recent months, but yeah,
I know.
we'll make it happen. But I did.
I am definitely going to fix that in 2026.
We'll do that. But I did, as I alluded to before you came on, get to sit down last summer and have breakfast with Dr. Jeffrey Miller, who is the brains behind the science here. We're going to talk about him in a few minutes. But I want to start, I love to talk in themes. You know, we can get lost in the weeds and talk about different scientific terms and lose people. What we're here for today basically is finding a better mousetrap, and specifically to treat cancer and hopefully reverse cancer in people. I loved something I heard you say on another interview recently, which I couldn't agree with more, looking at how my mother spent the last 25 years on and off of her life. You know, our treatment for cancer as a society is cut and burn. You talk about chemotherapy, radiation, and I love the term that you used. It's barbaric.
It is. Absolutely it is.
And the science that Dr. Miller came up with, as I got to get my head around it, and I've looked at it a few times and visited with you, visited with him, it's remarkable in a way in its simplicity. Theoretically, I know scientifically it's not that way.
Yeah.
But basically it takes, and I showed one of your videos before we started, so I won't belabor it, but it's really neat how you're able to make an NK cell, a natural killer cell, which is just a white blood cell in the body that fights cancer, how you make it far more efficient, but also do it in a way that some of the traditional cancer therapies that will cause toxicity problems and you got to stop the therapy or slow it down, that you've mitigated a lot of that. It's just really cool technology. So give us the kind of dumbed down 30,000 foot level view, Mike, of how this essentially works as best you can.
Delighted to, Chris. So just to recap then, and to make sure that the audience get the point, the current treatments for cancer, depending on what type of cancer you are unfortunate enough to get, have been around since just after the Second World War. So they've been around for 80 years. And it's incredible to think that in 80 years of medicine, there hasn't really truly been a huge step forward in finding a more humane, less barbaric treatment. Because, chemotherapy is just poisoning your blood, which ironically actually kills your NK cells, your white blood cells in the process. It kills everything basically. That's what it's designed to do. But the thing that you need, of course, are your NK cells, your natural immune system. And if you have chemotherapy, that basically wipes it out, which is why very often, if you're being treated, for example, leukemia or whatever, they'll say to you, you can't go out for 2 weeks after you've received an IV infusion of chemotherapy because you don't have any immune system and you could get the flu or a bad cold and it could be really catastrophic. So if it's not chemotherapy, then it's burning your flesh with radiation. And if it's not that, it's cutting your flesh with surgery to try and cut out the cancer. And of course, the problem with cancer is it moves around your body in through the blood system, but it also can get into your lymph nodes. It can move around. So simply thinking, often mistakenly, that it's localized into one particular area where they find a solid tumor is sometimes I feel a bit naive. So our technology is basically looking at is what has gone wrong with regard to the body's immune system and trying to fix that. So obviously with cancer, what happens is you have a normal cell that somehow turns bad, it mutates, it becomes cancerous, and then it splits it and mutates and it just keeps multiplying, splitting again. So suddenly two cancer cells become 4, become 8, become 16, become 32, and so on. And for whatever reason, the body's own natural immune system is not able to either detect and or kill and keep killing the cancer. So you effectively have three issues there. One is that you activate the body's natural immune system, the natural killer cells or NK cells. So activation, then you have to have proliferation so that they're in sufficient number that they can attack and overwhelm the cancer, whatever type of cancer it is. And then the third and final, but equally important part of what needs to happen is there needs to be sufficient persistence. So the NK cells, natural killer cells, have to be sufficient in number and keep killing until all of the cancer is eradicated. And that, I'm very happy to say, is exactly what our molecule does. So it's an NK engager. It activates the natural killer cells, it proliferates them, and it gives them persistence.
Well, and I loved when I first learned about the science or started to, Mike, the characterization that was made is it turns your body's NK cells into serial killers.
Yeah,
They don't
that's a
kill
good
one or
one.
two cancer cells and they're gone. They become far more efficient. So, and this is all based on what's called the trike technology that Dr. Miller at the University of Minnesota developed, and you know I'll put it up here again, folks. Some of his bona fides, his background. One of the leading cancer researchers, literally on planet Earth, is
Yep,
behind this, is behind the science, and it was remarkable what I learned from him. And he dumbed it down pretty good for me when we had breakfast, you know, to really get my head around it. So you're basically making the NK cells sturdier. They kill more
yep,
cancer cells, and that's one neat thing in the way that it happens. But the other thing that also really rang true with me, and again, watching my late mother once upon a time, and so many others. I mean, everybody watching this has lost somebody to cancer, I dare say. And a lot of the things that happen with the typical treatments out there today is you have a toxic response. Even if you're doing something that makes half a amount of sense, you got to slow it down or stop it because if you go overboard in your body with those NK cells, you have a toxic reaction, and now the cure is almost worse than a disease. So how do you avoid that part? 'Cause that's really cool science.
so that's a very interesting point. So this is where I'd make a very clear and important distinction with regard to our technology
Okay.
and T cell technology. So T cell technology, and I'm sure you already know this, but in very simplistic terms, you take the T cells out of the body, you reprogram them, and you put them back in. And as everyone knows, with certain T cell technology, part of the problem is it often leads to toxicity. In relation to what we are in, and there's two methods, I'll come on to this in a little bit, with regard to how we can administer our drug. One is we can give it through an IV, an intravenous infusion, and we often do that in blood cancer because people who've got acute myeloid leukemia are used to going into hospital, being hooked up to an IV, and then given chemotherapy. So obviously, we're not giving chemotherapy, and therefore you don't need to give anti-sickness drugs or anything with our drug because that isn't an issue. But what happens is we can administer it through the continuous infusion, which also gives you the benefit of at any point in time being able to switch it off if you need to. Not that we've ever needed to. And the second thing is, and I'll come on back to this in more detail in a moment, but we're going to be commencing in 2026 a second phase 1 clinical trial for solid tumors, and that's going to be administered via subcutaneous injection. So just basically like people, take their fat reducing injections, which they administer themselves or diabetes or whatever. You just stab yourself either in the leg or the hip or the stomach, and then through the body's natural fat, it's a slow release. And the other point I would make is our drug is actually so it's an NK cell engager. So it
Right.
engages the natural killer cells, and the other phrase I use, yes, it's a serial killer, but it also our molecule effectively turbocharges the natural killer cells. So in terms of proliferation and persistence, but because it's a protein, we have not seen any issues with regard to toxicity.
And that's key. And I know that in a lot of areas of science right now, I'm following a couple of companies working on Parkinson's, Alzheimer's, and whatnot. They have determined that it's a protein makeup or a protein at the core of a lot of what goes on in the body. And
Yeah.
I have a family member that has an offshoot of it's called a rare metabolic disorder, my oldest son, and we found out
Okay.
over time that it's proteins in his system that
Yep.
need to be augmented. I mean, it's I hate finding these things out the hard way like this, but it is remarkable that we've got some very bright minds, in this case, Dr. Miller. So you mentioned the hard tumor trial is going to be coming up hopefully early next year, but you've been putting out some news recently on a current trial for
Yep.
GT 3650 for bloodborne disease, and you've had some success with that so far in the phase 1 trial.
So, yeah, we have had a lot of success actually, and just to give your investors a little bit of background, so we did conducted a phase 1 trial for blood cancer in 2021.
Right.
We dosed 12 patients. So the way these things work, just again to explain it in a little bit more detail, is the FDA are, and rightly so, very prescriptive that you start on an extremely low dose because they don't want anyone being harmed, quite rightly. So you don't really see anything of interest until you get escalated to the higher dose level. So for the first 4 patients, we didn't see anything, which was to be expected, and in the 2021 trial.
Right.
But on patient 5, there's a reduction in bone marrow blast count levels of 33%. Patient 7, there was a bone marrow blast count reduction of about 60%. Patient number 9 was bone marrow blast count reduction of 65%. And then patient 11, there was reduction of 50%. So the interesting thing is that having conducted that trial, we were kind of at a crossroads because it made sense having what we'd seen with regard to significant reduction in bone marrow blast count levels to go straight to phase 2. But our dilemma was that we had actually developed a significantly improved what we now refer to as a second generation
Right.
trike. And the significance of why it was better is because in our preclinical trials and experiments, it was between 10 and 40-fold more potent, which is hugely significant. And it was vastly improved because we use what we call nanobody technology, which in simplistic terms just means it's infinitely smaller, tiny fragments, which means that it's easier to bind on to the cancer cell, and gives it significantly better binding affinity. And also, what we refer to as our secret sauce, the interleukin 15. That was a camelid type of IL-15. And again, that was much improved over the previous type in the 2021 trial. So you're right. We're now halfway through exactly our second generation phase 1 trial
Yeah.
for blood cancer, so for AML and MDS. And we have in the first, again at the low dose levels, we have seen a lot of interesting things in terms of activation. But we're now at the point, we're on the precipice of the second half of the trial. So we're now at the uh, dose levels as we were in 2021, where we're believing that we're hopeful that we're going to see some significant efficacy with regard to reduction in bone marrow blast count levels.
And that's really what the goal is. I mean, for folks who don't understand the whole trial process and to simplify it, when you're in trials with patients, you're looking for two things. Is it safe? Does it work?
Yep.
Okay.
Yep.
Safety issues pretty much has been settled. Uh,
Yep.
and then now we're looking at efficacy. And again, to get the efficacy you want, you're following protocol and you're bumping a dose up each new set of patients that you dose to get that. And so, and on that, you're going to be putting out some additional results on these later cohorts that you're enrolling or about to enroll with some of them early in the new year.
Yes, that's exactly right. So we're currently treating patients 7 and 8. So that's cohort 4.
Okay.
And cohort 3 was at 5 micrograms per kilogram of body weight. And cohort 4, this is where we get into significantly higher dose levels. It's 10 micrograms per kilogram of body weight per day. So it's literally double the amount that was given to cohort 3. And then in cohort 5, it'll be 25 micrograms per kilogram per day. Cohort 6 will be 50, and cohort 7, if we need to get there, would be 100 micrograms per kilogram per day.
Okay.
So we're really getting to significantly higher levels, and we will be most definitely releasing the data from those doses of the patients and those cohorts in early 2026.
Neat. And assuming everything goes well and you get the results you want, once you're through with that phase, what would be next for 3650?
So for 3650, the obvious thing would be to move to phase 2. And I'd also just highlight to you, Chris, that I don't know whether you recollect, there's no reason why you would, but in 2021, with our first generation molecule, the results were sufficiently good that we got what's called orphan drug status,
All right.
and we're hopeful that we will get that again. And that effectively, put it in very rough layman's terms, means that you have the ability or you can have it fast tracked. It'll be a easier, quicker process to get approval ultimately. And that's something that we're hopeful of also.
Oh, for sure. So, and then after that, you alluded this a few minutes ago, you're also going to be filing with the Food and Drug Administration early in the new year. I believe you said an IND, which is a paper for investigational new drug is exactly what that means.
That's
And that
exactly what that means.
and that will be for the part of the platform that specifically deals with these hard tumors, correct?
Yes, so that's correct, Chris. So the just again, just to explain things. So the the science that Dr. Miller has invented is a platform technology.
Right.
And that is very, very important. I can't stress enough how important that is because if you look at big pharma and the way that they approach companies like ours who have a platform technology, they tend to pay a lot of money for them. So the example I'll give you is Kite Pharma, which was acquired by Gilead for $8 billion. And the reason why big pharma like companies with platform technology is because if it works in one trial, it's highly likely to work in a second trial,
Right.
and it's also likely to be easier to get it through the different hurdles or over the various hurdles with the FDA because they were potentially more comfortable with regard to the fact that it's very similar to some of the other types of molecule. So for our molecule, we call it a trike because it's a tri-specific um, killer engager. So tri-specific simply means it's got three functional parts to it, to the
Right.
molecule or the protein. There's the target. So for blood cancer, the target is CD33. For solid tumors, it's going to be B7H3, and for autoimmune disease, it's going to be CD19. And we've spent a lot of work and a lot of research working out what are the best targets. So for solid tumors, pretty much all solid tumors highly express B7H3. So that's an excellent target to go after. So there's the target. Then there's the the interleukin 15, which I mentioned earlier, and then there's the receptor, which is CD16. So there's three parts. So when you want to treat blood cancer, you just swap out the target, CD33 for blood cancer. When you want to go after solid tumors, you take out the CD33 and you replace it with B7H3. And similarly, with autoimmune disease, you replace it with CD19. So it's really important to say that we're very excited with regard to the fact that it's a platform technology. And then going back to your question, which you mentioned in relation to solid tumors. So we're going to do a basket trial. And that means that we're going to go after all of the main solid tumors. And by that I mean, and so the most deadly and prolific. So head and neck cancer, lung cancer, um, ovarian cancer, bladder cancer, pancreatic cancer, and of course, breast cancer and prostate cancer.
Yeah.
And just to explain to your investors how prolific some of these are. So it's estimated that the solid tumor worldwide market is worth something between 280 and $400 billion a
Wow.
year. And part of the reason for that is that depending on what where you look, you'll see that unfortunately breast cancer affects 1 in 8 women a year, and prostate cancer affects 1 in 8 men also.
Wow.
And it's also estimated, um, because we're all living longer,
Mhm.
that the population of the US by 2030, 20% of the population will be over the age of 65,
Wow.
which effectively means, rightly or wrongly, that more people are going to get cancer. And that is why it is so important that we find a more humane
No question.
solution for this that works. And that's
So,
exactly what we're trying to do.
yeah, and again, folks, especially if you've got a little bit of medical aptitude or you're intrigued by what Mike has been talking about, make sure again that you go spend some time at their website. They got a very, very easy to navigate site, in my opinion. I mean, it doesn't overwhelm you that much, but it is educational. I mean, I guess the way to simplify it, and tell me if I'm too hokey on this, Mike, but you might have a great Cy Young Award winning pitcher. He's the trike. He's the
Yeah,
foundation. And sometimes he's got to throw a curveball. Sometimes he's got to throw a changeup. Sometimes he's got to hit you with a fastball, depending on what he's facing. And that's kind of what you're doing with the different cancers, is the way you
yeah,
explained it.
exactly right.
Yeah.
And we're we and by the way, so for the solid tumor trial, we'll file the IND in early part of 2026, as you mentioned, and the trial will start hopefully in the first half of 2026,
Yeah.
and we'll go after all of those big solid tumors. But commercially, if we get just one of those right, it will be completely life-changing for
Oh,
millions of people around the world, but also for the company, it will be beyond significant. And then, of course, in 2027, we're looking to file a third investigational new drug application with the FDA, and that will be for autoimmune disease. And the autoimmune disease space is estimated to be worth somewhere between 115 and 120 billion dollars per annum.
wow. That's something. As we start to wrap up, Mike, I want to address the company's funding
Yep,
because one of the things, I mean, as I said before you came on, what's past is past. You look at something like this, whether you already are a shareholder of GT Biopharma or saying, boy, do I want to take this risk? You look at all the different ingredients and ask yourself, would I buy this stock today? And I think it's a great speculative stock to buy. Speculative still is, of course, until you have more progress. But now you've been able to check a couple of boxes. One is to have some early trial success with 3650. But the other thing is financially. You guys reported in your Q3 that as of the end of September, you had 2.6 million cash on hand. That if
yep,
that's all you had would get you through the early part of the year. But you also have a couple of key investors who
correct.
have committed to a minimum of a $20 million credit line, which is essentially kind of an at the market kind of stock issuance. So as you need the money for trials, they pay around what the current market is. And one of the reasons I have my own renewed confidence in GT Biopharma is I know one of them, and he's been in this thing since day one.
He
And
has.
he loves the science and he's committed to it. And so that makes me sleep a lot better at night
Yeah.
with this. So having that support from a couple of your key investors, I think really is the corner turner, if you will, because last summer when I was visiting with Dr. Miller over breakfast, he was very candid and he said, "Man, we got a tiger by the tail. We could be doing great things with this, but we got to get funded. We need the money."
Yeah.
You
Yeah.
don't get anywhere if you don't have the money to go through trials. And now it doesn't seem like that's going to be an issue anytime soon. So that's great.
That and as you can imagine, Chris, for me, the role of executive chairman and CEO, I'm not the science guy. I'm the guy. I'm the business guy.
Yeah.
So if we're going to do some sort of deal with big pharma at some point, I'll be the guy who negotiates that
Yeah.
deal. My background, of course, is I was a mergers and acquisitions partner in a global legal practice. Um, I was a senior equity partner in that firm, and I sat in the management board, and I did that role for 25 years.
Yeah.
So that's my bread and butter. It's what I do, and I'll be ready to go when that moment comes. But I'd also say, from the way I view the world, um, the first job of work that I perceived was super important for me was to cut all of our overheads
Yeah.
as low as they could go. And that is exactly what I did. And that's what I've maintained a very sharp pencil for all the way through this. Because it's the thing that I can control. The other bit I
Yeah.
can't control, you know? So that was equally important. And then the third bit is, of course, the funding for the company. And unfortunately, there are lots of pharma companies that may have had good science, but nobody ever found out because they ran out of money
Yeah.
because they were either over optimistic or they weren't very good at cutting deals or whatever. And for me, the most important thing is making sure that we're adequately funded to get to where we need to get through the trials, to do all of these solid tumor trials, and then hopefully get to a point where we ultimately get approval from the FDA. But normally, the way it happens, of course, is big pharma come and knock on your door before you get there. But hey, you can always structure a deal in these
Sure.
circumstances.
Well, and hey, from folks today, as Mike and I are talking, under a $10 million market cap, for science that could be worth in a multiple billions of dollars one day. And I think that's a pretty nice potential risk reward, especially when you've got the funding now in place that wasn't there that long ago. So, Mike, congratulations for sticking it out. I know that when you
Thank you.
came in, you had a hell of a cleanup job, but now
I
things are
did.
back on the upswing. And I'm excited for what you're doing, and looking forward to seeing you sometime in the new year. And I'll be back and see, I don't know when it's going to be, but I'll be visiting Dr. Miller next time I'm up in the upper Midwest as well again.
That would be great. And I can't tell you how excited we are for 2026 with everything that we've got going on. So
Yeah.
I'm being fully funded. That's
Yeah.
super important. So roll on
Overdue,
2026.
yeah, overdue, encouraging story. Mike, thanks much. We'll see you again down the road.
Many thanks, Chris. Really enjoyed spending time with you.
Take care.
For much more of the National Investor's cutting-edge content and actionable investment opportunities, make sure to subscribe to Chris's free content at nationalinvestor.com. And don't forget to like this video and subscribe to this channel too. Thanks for tuning in, and we'll see you next time.